Do you think analytical method development is just a technical step? In reality, it's one of the most critical phases that determine your pharmaceutical product's success with regulatory authorities. I'm Dylan, AI expert at Cobalt Research, and in this video, we'll walk through how to properly develop an analytical method, from method design to full validation. Hello everyone, welcome to Cobalt Research channel. Before any pharmaceutical product reaches the patient, or even gets regulatory approval, one crucial phase must be done with absolute precision: developing and validating the analytical method. It's not just a routine procedure or a checkbox in your submission file. It's the backbone for evaluating product quality, stability, purity, and even efficacy. The surprising part? A large number of regulatory rejections stem from analytical issues, not formulation or clinical content. We'll explore how to develop a method that's scientifically sound, how to ensure it's reliable, and how to avoid the common pitfalls that waste months of effort. Let's break it down step by step. Step 1. Method development. We start by defining what exactly we want to measure. Is it the active ingredient, degradation products, manufacturing impurities? The key here is to understand the compound's characteristics: solubility, stability, interaction with excipients. Then we select the appropriate analytical technique: HPLC, UV, GC. Each has its advantages based on the compound's properties. For instance, if we're working with a light-sensitive drug prone to degradation, we might use HPLC with UV detection at specific wavelengths to separate the parent compound from its degradants. We also define the chromatographic conditions: column type, mobile phase, flow rate, temperature. Every parameter affects resolution, accuracy, and retention time. But the goal isn't just to make it work in the lab. The method must be repeatable, clear, and practical for future QC labs. Step 2. System suitability. Before any actual testing begins, you must confirm the system is working correctly. We do this through system suitability tests, such as resolution between compound and impurities, theoretical plates, tailing factor, repeatability, RSD%. If these values fall outside the acceptable range, it signals a problem with the column, mobile phase, or instrument, and it must be resolved before continuing. Skipping this step can produce misleading results, even if the data looks fine. Step 3. Method validation. Validation is where we prove the method actually works and is fit for its intended purpose. Depending on the method type, assay, impurities, dissolution, we follow ICH Q2 guidelines and validate the following parameters. Accuracy. Does the method return the true value? Precision. Are results consistent across replicates? Specificity. Can it distinguish between the active ingredient and impurities? LOD/LOQ. What's the lowest detectable amount? Linearity. Is the response proportional to concentration? Robustness. Does the method tolerate slight changes? Let's say we're validating a method for an API in a capsule. We may use 6 different concentrations and calculate scent recovery. If recovery is between 98 to 102% and RSD is under 2%, the method is acceptable. But if an impurity elutes close to the main peak, you must prove specificity, showing that each compound is clearly separated, even in the presence of degradants. Final step: why it matters. A poorly validated method equals unacceptable data, and a submission could be rejected entirely for a single analytical issue. Many companies lose months of progress because their method validation is incomplete, inconsistent, or poorly documented. That's why method development isn't just about running a machine, it's a core part of your regulatory strategy. At Cobalt Research, we support your team from initial method design through lab performance review and deliver complete validation reports aligned with EMA and FDA expectations. The right analytical method proves product quality, supports stability, and answers regulatory questions. Start with solid scientific development, fully validate your method, and document every step. If you're developing a new product or reviewing current analytical methods, we at Cobalt Research can help you do it right, from first trial to final submission. Thanks for watching, and don't forget to follow us for more expert insights that help you move your development forward with confidence.